The final thing to talk about with antibiotics is how do we test, okay? How do we test to see if a bacteria is sensitive to the antibiotic, meaning the antibiotic will kill it, or if the bacteria is resistant to the antibiotic, meaning it can still grow in the presence of the drug. And these tests are really important anytime we have a patient who the antibiotics are not working against the specific disease, or if we have a newly discovered pathogen and we need to find a treatment for it, we need to do these antibiotic tests. So the first test, and one of the most important ones, is called a Kirby-Bauer test, also sometimes called a disc diffusion assay. So the way this test works is you take a media, solid agar medium that the bacteria can grow on, and you create what's called a lawn of the bacteria, which means you spread the bacteria over the entire plate. All right, so you spread it's a spread plate and you're spreading the bacteria everywhere. And then you take these discs, these little discs that contain the antibiotic on them. They're actually just made out of a paper, like a cellulose paper. So you do different discs. You might do a disc with penicillin. You might do a disc with tetracycline. You might do a disc with vancomycin. And you might do a disc with chloramphenicol. So you pick some different drugs to test. And then you wait and see if the antibiotic kills off the bacteria around it. So for example, vancomycin is a very strong drug against gram-positive bacteria. So you might see this big space where the antibiotic kills off the drug. Tetracycline, a very broad spectrum antibiotic, so you might even see even a bigger space where the drug kills off the bacteria. And then penicillin, not so great, so maybe the bacteria still grows or even around the penicillin disc. After this, you measure, the diameter of the zone of clearing. Okay, so zone of clearing or zone of inhibition. All right, just like pictured here, you'll take a ruler and you'll measure the zone of inhibition. So if you have a really big zone of inhibition, like in this picture, a big one, it means the bacteria was very sensitive to this drug and the antibiotic has killed off a huge amount of the bacteria. Whereas if you see this really small zone of clearing, the bacteria were still able to grow around the drug, indicating the bacteria were very resistant to this antibiotic. So that's the first test, a Kirby-Bauer test. So our next test to talk about is called an E-test, which is also a type of diffusion test. It's kind of similar to the Kirby-Bauer test. But how this works is, again, you take a plate and you create a lawn of the bacteria. Okay, you spread the bacteria all over the plate, and you can see this is the bacteria here growing. Then you take a strip, okay, and this strip is coated in a specific antibiotic. And down here at the bottom, you start with a very low concentration of the drug. And then as the strip goes on, the concentration increases to a very high concentration of the drug. So at a very high concentration of the drug, it's going to be really good at killing off the bacteria around it. So you see this big zone of clearing. Whereas at a very low concentration of antibiotic, the bacteria are going to resist because it's such a small amount. So the goal of this is to find the minimum amount of antibiotic that's enough to kill the pathogen. So in this case, if you look around in this area here, this is around the minimum amount of the antibiotic it takes to kill the bacteria. All right, because you see there's still a little bit of a zone of clearing before you see that they all become resistant. Similar to the E-test, you could also do the same test, but instead of on a solid agar, you do it in a liquid broth medium. This is called an MIC, or a minimum inhibitory concentration test. So again, You put the bacteria into broth, liquid medium. The control would be no antibiotic. And then you add a little bit of antibiotic and you increase it higher and higher. And you find the minimum amount of antibiotic it takes to kill off all the bacteria. So you can see these cultures are very turbid, so the bacteria are still growing. But in this culture, the broth is very clear. So you can see this is the minimum inhibitory concentration, 1.6 micrograms.